Following the first lecture with the glycolysis, we now go on to the second part. So in this part, we will describe the actual glycolysis on the first stage. So the first stage is what is depicted here, and the third lecture will take care of the second stage, which is the stage down here. So in the first stage, we will convert glucose into two 3-carbon molecules, which is called glyceraldehyde 3-phosphate. So glycolysis is a way that cells oxidize glucose, and the glycolysis is part of a pathway that leads to the full oxidation of glucose. It can also lead to fermentation, as you can see on this slide. There could be fermentation into ethanol, which can be quite useful in some instances. It could also be fermentation into lactate. So lactate fermentation is what you experience when you run for the bus. So the lactate formation down here is what happens inside your muscles when you are running very, very fast and don't have any oxygen levels in your cells. So that's where you will find that kind of fermentation. Sometimes, the other times you will find it in lactobacillus, which is what we see inside yogurt, for example. Now, but today we will talk, in this one, we'll talk about how we turn glucose into 2,3 molecules. The first step in the glycolysis is a hexokinase. So the hexokinase, well, as it says, is a kinase. So it requires phosphate. And this is one of the key things about kinases. They utilize phosphate some way or the other to function. Mostly, they also regulate it by phosphorylation. That means that a phosphate group would sit on the kinase. So the first step here, we have the glucose, and via the hexokinase and a conversion of ATP into ADP, we will get glucose 6-phosphate. So that means that the 6 group, which is up here, as an alcohol in the substrate, will become a phosphate group in the product. So that's the first step of the glycolysis. Now, as we talked about before in other lectures, the glucose or the hexokinase works by induced fitting. So induced fit, as you see up here, is important in this situation, since if we did not have induced fit, the ATP would then be converted into ADP, but it will be via hydrolysis with water. So instead of having a water molecule that is then turned into ions, we can utilize the glucose and bind the free phosphate group that we would get with water and bind that to glucose instead of water. Now that is where the induced fit is very important. So the induced fit stops the penetration of water into the active site of the molecule. So where down here we have a regulatory site, we have a substrate binding site. That's also what we call the active site. And this is where we will bind the glucose and the ATP. As you can see up here, they bind into the active site. When they both are bound, we can get a hydrolation of the ATP into the ADP, and we get the phosphate then conjugated to glucose 6-phosphate. Now, if we have a lot of free phosphate in this, as you can see here, or not a lot of free phosphate, if we have a lot of ATP in the cells, then we also get a lot of glucose 6-phosphate. Glucose 6-phosphate then has a feedback regulation on the hexokinase, and that you see down here on the right part of the slide. You can see there is this site where we can bind glucose 6-phosphate into. In that situation, the enzyme changes conformation, and as you can see here, there's no longer room for ATP inside the active site. Now, this kind of feedback regulation is quite normal, and the binding to a secondary site of the enzymes is called allosteric regulation, and we'll get into that in other lectures as well. So allosteric regulation is a very crucial way to regulate enzyme. This figure also shows where this induced fit mechanism comes in. So you have this open mouth first on the left side that closes over the two substrates as just showed up here. So that's where the induced fits come in. Now, the hexokinase comes in a secondary form within human cells. So these are especially important in liver cells. And it's a form of the hexokinase that's called glucokinase. So glucokinase is an enzyme that is upregulated when we eat sugar in humans. So that means if we have a lot of sugar in our blood cells, the glucokinase is upregulated. And this glucokinase can be used for glucose monitoring in blood cells. So that's a way to do that. So what you see here on your right-hand side is that you have a graph where you have the glucokinase and the hexokinase. And as you can see, under almost all normal level, the hexokinase is at a steady state. It's up here having the same level all the time, whereas the glucokinase depends on the level of the glucose concentration. You see that it increases very nicely over the glucose concentration. So that gives two different Km values for the two enzymes. That means that the affinity of glucose is much higher to the hexokinase than it is to the glucokinase. But we can utilize the presence of the glucokinase in the bloodstreams to help with monitoring glucose, and especially for diabetes patients, that's very important. So these are two different forms of the same enzyme does the same reaction for humans in our cells. So the two differences mainly here is that you can see hexokinases are, it says here, in many tissues. It should actually says in all tissues, because hexokinases are also present in liver and pancreas. Whereas the glucokinase is only found in liver and pancreas. So here you see an example of heterologous or orthologous enzymes that are expressed differently throughout our body. So this is true for all mammalians, and is somewhere that that we can control these findings. The other specific parts on this slide is that hexokinase is inhibited by glucose 6-phosphate, whereas glucokinase is not. So that means actually at very, very high sugar levels, with high ATP levels, glucokinase will still function. Is that a benefit? Yes. So if we have a lot of sugar, we can turn it into a phosphate molecule that we can use later on. So the second reaction in this glycolysis is the phosphoglucoisomerase. Now, this is simply just an isomerase. So maybe you have seen on the overview that at some point we get fructose, and this is where we get the fructose. So it's an isomerization of the glucose 6-phosphate into fructose 6-phosphate. And what does that mean? It means that we open up here in this this ring here at this position. So the ring opens there, and the alcohol group is then on the 5th position over here. So that one, the oxygen here stays with this carbon over here. Then when we close again, instead of creating a 6-membered ring, we create a 5-member ring. That means we create a bond between the oxygen from before and the C2 carbon, which is positioned here. So that's how we have this isomerase reaction. These are not particularly regulated, but they're very useful reactions throughout any kind of sugar chemistry or sugar biochemistry reactions, I should say. Now the mechanism is seen as here. So basically the molecule opens, as I showed here in this position, opens up to a open 6-membered ring. The change between the aldehyde that you see here and the ketone group that you see over here is basically controlled via that the the two lone pairs, that the lone pair you have here makes a double bond in here that then pushes the electrons through the whole system, and that these two then go out here. So that's how these can be related to each other. It's a reaction that can go forth and back with inside the enzyme. So this equilibrium is really strong. So there's almost 50-50 percentages of the two molecules that you see here open chain form. Now the open chain form is not the most stable, as you also can see both on here from the glucose 6-phosphate close to the open form, and to the fructose 6-phosphate open form to the closed form. So both of those reactions are pushed towards the ring structure. So the ring structure has a lower energy level. So that's why that is preferred. So first of all, when we got this, we can then the enzyme pushes the reaction in this direction towards the fructose 6-phosphate. This next step is the phosphofructokinase 1. So the phosphofructokinase 1 is an important step in the glycolysis, is one of the key regulatory steps in the glycolysis. Why is that? That is because it uses ATP. So ATP is a really good way to regulate things. Now, I've written here that this enzyme converts your fructose 6-phosphate into fructose 1,6-bisphosphate. There's a little trick here in the naming of these molecules. I don't know if you recognize it, but it's called bisphosphate. So that's the B here stands for the bisphosphate. So that's instead of the diphosphate, it says BIS phosphate. So that means that we have two phosphate groups. So we have one here and we have one on the other side. But if it was a diphosphate like the ATP and ADP, the two phosphate groups would be conjugating and sitting together in one, but here the bisphosphate tells us that the two phosphate groups are positioned on position 6 and position 1 over here. So that's the naming difference between the bis and the di in this situation, and that comes through all chemistry or biochemistry. So if you see these namings, you know that if it's a bis, they're sitting on different carbons. So this is the reaction that takes place, and the allosteric regulation in this is that it's activated by AMP ADP. Now here I would usually ask you, why do you think so? Well, I can do that, but you will not be able to answer. So AMP and ADP are products of these reactions, but they are the negative, so to speak, products. So we want to have a lot of ATP when we run glycolysis. So that's why if we have a lot of ADP and AMP means we are low on energy levels, and thereby we need to activate this reaction. So that makes sense that these two molecules, AMP and ADP, activates the reactions. On the other hand, if we have a lot of ATP in the cells, well, then we don't need more. So why should we use it? So that's why ATP is an inhibitor of this reaction. So is the phosphoenol pyruvate, which is a further downstream product in the second stage of the glycolysis. So that's written here. So how does it actually work? Well, the phosphofructokinase comes in two different forms, as you can see here. Well, the difference is not very big, but as you can see here in the middle, let me just get a color you can see. So in here in the middle, you can see there's a slight change in the whole, and you can see that the whole conformation of the molecule changes slightly. So that means that the molecule, the enzyme changes conformation when it is phosphorylated. So this is where the kinase activity again come in. So if we have phosphorylation of these things, well, then we can phosphorylate and we have an active form, and dephosphorylate we have an inactive form. So that's the way the kinases are regulated. Usually it's regulated. We'll get back to the regulations later. The fourth step in the glycolysis is the aldolase. So the aldolase is a key step in the glycolysis. So why is it a key step? Well, aldolase is where we get the fructose 1,6-bisphosphate. Oh, I forgot to say that it was a phosphorylation, substrate level phosphorylation. before. So the fructose 1,6-bisphosphate, now that has two phosphate group, and what the aldolase does is basically take this enzyme, it cuts it in two like here, and then we get first the dihydroacetone phosphate is one side, and the second part is the glyceraldehyde 3-phosphate. Now, if we want to continue further on in the glycolysis, we need to have two glyceraldehyde 3-phosphate molecules. So that's the 6th and 7th step where we need those. So in this situation, we actually make something that we would call normally a byproduct. That would be the dihydroxyacetone phosphate. It's more or less a byproduct because it would not directly go on. But luckily, there are ways to measure that. So the reaction mechanism is slightly different than what we've seen before. So how do you cleave a molecule like this into, basically you can take an axe, of course, and see if you can hit, but that wouldn't probably work. So the enzyme, what it does, if you look up here in the top right corner at number 1, so what the enzyme does is that it having a base, which is a shift base. Here is the amine group here, as you see here, that can react with the aldehyde group of your molecule. So that's the C2 position you see in here. And inside the active site, at the same time, it forms a hydrogen bond down here between a carboxylic acid group and one of the alcohols in your fructose. Now this stabilizes it. It also releases a water molecule up here, as you can see down here in this. So this in the second step, you can see that the water molecule is or the oxygen is no longer part of the molecule. So now we have a C double bond to the nitrogen. We still have this hydrogen bond down here, and that we can shift into this to make shift electrons into that bond, and then we can steal the hydrogen and then put the electrons back into the molecule like this. So in the third step, what happens here is basically that while we push in two electrons from the protons here on the right hand side into the molecule here, we at the same time then get a surplus of electrons around this carbon, and then we push those electrons from this bond up here. That means that this the CC bond here between the C3 and the C4, that breaks, and we get a release of the glyceraldehyde 3-phosphate, as we see down here. So that leaves us with this enamine intermediate here in the 4th position between 3 and 4 over here. So this enamine intermediate is not really something we can go forward with. The enzyme then do an isomerization via the acid group where we had before. You see down here the carboxylic acid down here. They protonated the shift base, and by adding water to the whole reaction, we get a release of the dihydroxyacetone phosphate, as you see down here. Now, this is one of the final products, and this is the final product down here. So in this way, it looks like the glyceraldehyde 3-phosphate is the byproduct, and the dihydroxyacetone is the true product. So that is to some extent true, but we cannot really use the dihydroxyacetone phosphate. So therefore, evolution has luckily designed a isomerized enzyme that can deal with this. So we have the dihydroxyacetone phosphate here, and that can isomerize via the triose phosphate isomerase enzyme, which is the 5th step of glycolysis, into glyceraldehyde 3-phosphate. Again, this is a step where we can play around a bit. So in different conditions, we can have different settings of these two things. Now, this stage also ends the ATP investment. So this is the first part of the glycolysis that we invested 2 ATP molecules into 1 mole of glucose. So at this stage, we have so far invested 2 ATP. The isomerase, well, a bit like the enidol intermediaries over the aldose, utilizes that we have shift bases around nitrogens. As you can see here, we can create one here to push the isomerization of the two molecules around. Basically, if you follow the electrons from the carboxylic acid on the glutamate 165, you can see they have them coming out here and making a hydrogen bond to this hydrogen that is then released over here. So we break a bond here. Then we can have an attack from the enediol over here to attack into here. And we can get electrons back into the oxygen here. Now that gives a negatively charged oxygen you see down here in on your down right hand corner. That enables us to push the electrons around and create a new CH bond here so that we get this molecule that you see over here on your left hand side. And this is basically the glyceraldehyde 3-phosphate molecule in the end. So that's the way most isomerases work, basically by first having either having a carboxylic acid attack onto your molecule or having some kind of amine attack onto your molecule as you have here on your right side. So that's the two ways that these usually work. Okay, so that concludes the glycolysis first stage. So we basically have 5 enzymes. First, we have the hexokinase that phosphorylates the glucose into glucose 6-phosphate. The second step is an isomerase that isomerizes glucose into fructose 6-phosphate. Then we have the phosphofructokinase 1 on the third step that phosphorylates the fructose into fructose 1,6-biphosphates. And then we have the aldolase that cleaves the fructose into dehydroxyacetone phosphate and glyceraldehyde 3-phosphate. And the final step is basically an isomerase that takes the dehydroxyacetone phosphate and isomerizes it into glyceraldehyde 3-phosphate. Overall, you can see here there are 2 important observations here on your right hand side in this table. That is that the first step, you see that here, has a very negative delta T value, and so does the second step. What does that mean? That means that these 2 steps are virtually irreversible. So you need a lot of energy to drive those as depicted here. If you were to drive these in the left direction, you would need a lot of ATP to drive that, a lot of energy in the form of ATP drive that reaction. So those 2 steps is virtually irreversible. That's an important just to keep note of when we're going to talk about the glycolysis in the next sets of lectures.